The cells of all living creatures contain two types of nucleic acids, deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. Like a master blueprint for the construction of a unique living organism, DNA molecules contain the genetic information necessary for all the inherited characteristics of any particular organism.
RNA molecules assist in the synthesis of an organism's proteins by reading and translating the genetic codes of chromosomal DNA.
In human beings, these genetic codings and the proteins produced by them determine nearly everything about us, including physical attributes such as our height, our hair color and musculature, how we digest our food, whether or not we will have diabetes, even our general mental health and how long we are likely to live.
To understand protein synthesis, we need to familiarize ourselves with the structures and functions of DNA, mRNA, and tRNA. This is a model of a DNA molecule. This molecule here is M, or messenger RNA.
And this complex model here is a representation of T, or transfer RNA, complete with its corresponding amino acid. Both DNA and RNA are information polymers, which are composed of linear sequences of smaller elements called nucleotides.
In DNA, the sides of the nucleotides are composed of phosphate and the sugar deoxyribose, while the rung portion is composed of either cytosine, guanine, adenine, or thymine. RNA is similar but not the same. The sides are composed of phosphate and a different sugar, ribose, while the possible nitrogen-based molecules are cytosine, guanine, adenine, and uracil, with uracil being substituted for thymine.
To complete a rung of the helix, the nitrogen-based molecules must be paired with each other. Only certain combinations are possible. These combinations are called the base pair rules. In DNA, adenine may only join with thymine. And guanine with cytosine. In RNA, the combination of guanine and cytosine remains the same, while adenine now combines with uracil. These unique and exclusive base pair combinations hold the key to the perfect, exact, and identical production of the unique protein combinations necessary to any particular organism.
This is how genetic continuity is ensured. For a living cell to synthesize proteins from the master blueprints contained in DNA, two processes are required. Transcription, which involves creating an mRNA template from the DNA master, and translation, which involves the manufacture of specific proteins by the mRNA and tRNA molecules. Transcription occurs in the nucleus of a cell. The DNA molecule creates an mRNA molecule by first unzipping its helix along the hydrogen bonds of the rungs. It then assembles a new mRNA molecule using free nucleotides present in the nucleus. The initiation of the mRNA synthesis is specified by a nucleotide sequence called the promoter, and is concluded by another sequence of nucleotides called the terminator. Because of the base pair rule, only one unique mRNA molecule may be produced from any given section of the DNA molecule. Guanine always bonding with cytosine, and adenine always bonding with uracil. To begin the translation process, the newly created mRNA molecule migrates out of the nucleus into the cytoplasm. Here, the template will bond with and be read by a ribosome, and the synthesis of a new protein begun.
How is is information in the mRNA molecule used to produce an amino acid polypeptide, or protein. Let's look again at the mRNA molecule. The nucleotides in an mRNA molecule are arranged in groups of 3, called codons, or triplets. These groupings contain the genetic instructions from the DNA molecule, which indirectly specify the eventual arrangement of the particular amino acids in a polypeptide chain. Because each nucleotide in the codon can have one of 4 possible nitrogen bases, cytosine, adenine, guanine, or uracil, there are 64 combinations possible for each triplet, 4 times 4 times 4. In the cytoplasm of a cell, there also exists another form of RNA, called tRNA, or transfer RNA. These molecules, which have also been assembled by the DNA molecule, are designed to attach themselves to one of the 20 or so different amino acids available to them in the cytoplasm. Like mRNA, tRNA molecules are also composed of a number of nucleotides. Only 3 of them are significant to the translation process. This group of 3 nucleotides is called an anticodon, and each corresponds to complementary codons in the mRNA molecule. Again, there are 64 possible combinations for each triplet.
The ribosome now coordinates the coupling of tRNA anticodons with mRNA codons, and in so doing, links the amino acids, which are attached to the tRNA molecules. This produces the polypeptide chain. It is possible for hundreds of ribosomes to read the same mRNA molecule at the same time, each one at a different stage of protein synthesis.
Actual protein synthesis occurs in 3 stages. In the first stage, the tRNA molecule and its amino acid binds to the small ribosomal subunit. This complex then couples with the mRNA molecule, codon to anticodon. Finally, the large ribosomal subunit bonds into place. At this point, the second stage begins. In this stage, amino acids are added one by one to the original amino acid. As each amino acid bonds to the other, it gives up its bond to the tRNA molecule to which it was attached. These tRNA molecules will eventually retrieve still more of their specific amino acids. The final stage, called termination, occurs when a termination codon is reached. A protein, called the protein release factor, bonds to the mRNA molecule, causing the dissociation of the ribosomal subunits and the freeing of the polypeptide chain.
A new protein has been produced. This new protein will go on to perform some vital function in the organism. If only one of the codons in an mRNA molecule has been altered, the protein it produces will also be changed. This is how genetic abnormalities occur.
From this information, can you describe the process by which genetic diseases occur and why they are hereditary?